Przewalski's horse might be the last truly wild horse. It diverged from the domestic horse somewhere between 35,000 and 54,000 years ago. But back in 1996, Przewalski's horse was all but extinct. Since then, conservationists have been working to bring these horses back from the brink. And thanks to their efforts, there are now roughly 1,300 of them running wild in the plains of Mongolia and China, along with another 1,000 or so in zoos and breeding centers around the world. But there's one big problem. All of these horses have been bred from just 12 to 16 individuals, so there is not a lot of diversity in their gene pool. And that brings with it problems like inbreeding, reduced fertility, and shorter lifespans. To try and deal with that lack of diversity, scientists have cloned horses to release into the population using DNA collected in the 1980s from a single horse. One foal, named Kurt, was born in 2020, another named Ollie was born in 2023. But how did a dead horse become a dad? Well, through the science of cryopreservation. And freezing DNA might just be the kind of scientific insurance policy we need for putting extinction on ice too. If we're okay with the ethics of it all. In the 1970s, scientists realized that freezing genetic material could help in their conservation efforts. After all, it's literally preserving a little piece of an animal that you could then use to make more of them, or, like with Przewalski's horse, keep the population genetically healthy. Over several decades, cryobanks popped up all over the world. They store not just DNA, but embryos, semen, and live tissue, which can be used for research or for breeding. But of course, this isn't just as simple as plopping a chromosome ice cube into your regular freezer and calling it a day. Cryopreservation is an exact science. Samples are collected and then frozen at around a brisk minus 196 degrees Celsius. They're usually also treated with some kind of cryoprotectant, which stops ice crystals from forming that could warp or alter the structure of the biological material, effectively killing it. At those temperatures, it's like the cells are suddenly frozen in time. All of the chemical and physical processes going on inside suddenly stop, but basically exist in suspended animation, ready to jump back to life when the samples are warmed up again. And when they are, the samples can be used for all sorts of science and conservation efforts. For example, the largest animal cryobank, the San Diego Zoo Wildlife Alliance Frozen Zoo, used thawed cheetah sperm to fertilize a cheetah egg from one of their cats to make an embryo. Cheetahs can still be notoriously difficult to breed in captivity, and even artificial insemination hasn't been all that successful. So, transferring ready-to-go embryos might be the best way to get the animals' numbers up. Researchers from the American Museum of Natural History in New York City and Tulane University in New Orleans have also used DNA from cryobanks to create libraries of genetic barcodes, sections of DNA that are unique to different species. For example, in 2010, they created such a barcode for different marine turtles who look remarkably similar, and they could use this barcoding to tell them apart. That's practical in a conservation sense because that barcode could help them track the illegal trade of the turtle's eggs or meat by tracing the item back to its geographic origin. What's more, the samples in cryobanks can be used by scientists to study the population genetics of species at risk of extinction or test out new assisted reproductive technologies. But for all of their conservation catalyzing triumphs, cryobanks are not perfect. For one, even though there are many cryobanks around the world, they could be more representative of all of the weird and wonderful wildlife out there. For example, a 2023 study by Andrew Mooney and colleagues did a kind of cryobank stocktake of the frozen zoo in 2019 and found that while the frozen zoo contains material from 965 different species, that only includes 5% of the reptiles, mammals, birds, and amphibians listed on the IUCN Red List as threatened. Which is not bad, but with a bit more sampling from zoos and aquariums, the authors of that study calculate that this percentage could increase to 16.6% or include an extra 707 threatened species. And of species listed by the IUCN as extinct in the wild, the frozen zoo has genetic samples of half of them, but based on their proposed sampling from zoos, that could increase to 91%. And even with the best kept and used cryobanks, there are still a whole host of ethical issues to consider. For one, how do researchers choose choose what gets stored and saved. Partly this might come down to what's available, like the stocktake study suggested, but it also depends on feasibility and how much value we humans place on keeping certain species, which often means picking the charismatic ones. And for another, who gets to use the precious samples that are stored in cryobanks, and who decides? Is it the people for whom the animal is culturally significant, the biologists trying to save a species, those in charge of the banks themselves, or like some other independent body? Then there are questions of whether we should be bringing animals back from the brink at all. In an essay published in 2013, philosophy professor Ronald Sandler pointed out that the process of cloning can be pretty stressful for the animals and lead to health problems during development or the suffering of the clone and the surrogate that carries it. He also noted that creating new individuals doesn't address the root cause of why these animals are endangered in the first place. Things like habitat loss, climate change, or pollution still exist once a clone has been made. When it comes to Przewalski's horse, cloning is a bit of a last-ditch lifeline, and there's at least some effort to make sure conservation efforts address ongoing threats to the species as well. The animals and the areas where they live are now legally protected in Mongolia, and efforts are underway to tackle problems like illegal mining of those areas and human interference. Since 1996, the species has gone from extinct in the wild to critically endangered, and as of 2011, endangered. And the goal is now for the cryobank horses, Ollie and Kurt, to become breeding stallions. The hope is that they can sire more horses that will be released into the wild and keep the species going for years to come.
There's a new predator in the ocean depths. This beast lurks around Atlantic coral reefs, looking different than any sea creature you might have seen, and has especially effective hunting habits. Its prey? Lionfish, one of the most destructive fish in that neighborhood. So what are these new apex predators? They're robots, piloted by scientists deployed to restore balance to coral reef ecosystems. Coral reefs benefit both land and sea alike. They support around 25% of all marine life and protect nearby shorelines. But these vital ecosystems face many threats, both natural and human-caused. These threats can disrupt the delicate balance between organisms, potentially throwing the ecosystem into chaos. And in the case of Atlantic coral reefs, lionfish are a big one. That's where these robots aim to help. Sometimes it takes a new kind of predator to tackle an old one. Now, the reason lionfish spell trouble for Atlantic reefs is that they don't belong there. They're invasive, meaning they aren't native to those waters and are destructive to the sea life there. Lionfish are native to Pacific and Indian Oceans, where they're eaten by other predators, like large moray eels, so they don't cause problems for those ecosystems. But without natural predators to keep their numbers down in the Atlantic Ocean, they wreak havoc. They probably got into the Atlantic in the first place due to aquarium owners tossing their unwanted lionfish into the sea, making them a problem that we humans introduced. Shocker. That might sound like a reach, but lionfish can reproduce pretty impressively. They mature in 12 months and can spawn 2 million eggs per year. What's more, they're powerful predators, eating a wide variety of reef fish and crustaceans. If it's less than half the size of a lionfish, it's not safe. If that wasn't enough, lionfish have venomous spikes, providing effective armor against predators that aren't equipped to handle them. This means that their numbers and their destruction aren't kept in check by predation in the Atlantic. That is, until conservationists decided to step in. A team of conservationists and scientists developed a robot to enter the coral ecosystems as the new top predator of the reef, targeting lionfish exclusively. It is called the Guardian. It looks a bit like a long robotic tube with a camera on top and 2 metal arms sticking straight out in front of it. Each of these features serves an important purpose. The camera allows the operator to pilot the robot using a controller. With the help of an AI system that identifies, tracks, and positions the robot, the Guardian pilot duo approaches the prey and uses its metal arms to deliver a zap to stun the target, not kill it. Much like a robot vacuum sucks dirt and debris off the floor, the Guardian suctions the stunned fish into its capture chamber. The original robot can hold 10 lionfish at once, and it's designed to work fast too, potentially capturing 50 to 100 fish per hour. With enough usage, the robot fishing can add up. But because of the manual nature of the Guardian's first prototype, that would require a consistent presence of human pilots. Because lionfish are a popular fish for humans to eat, the developers of the Guardian debuted its prototype at a lionfish cook-off in 2017 to raise awareness about the invasive species and market the device to anglers as a solution to the problem. Since then, the Guardian has improved to hunt uncrewed, as well as carry more lionfish and run for longer on a single charge. The Atlantic lionfish dilemma is an example of how human influence can both harm and benefit an ecosystem. People introduce lionfish to the Atlantic reef ecosystems in the first place, but enough robot fishing can potentially restore balance. After all, we're animals too. And when done responsibly, one study published in 2022 suggested that humans can behave like any other creature in the ecosystem and act as top predator. This time with some robotic assistance.
You've probably heard of the Black Death, one of the most fatal pandemics in human history. Well, you might not know that the disease that caused it, also called the plague, is still around today, including in the United States, Madagascar, and Peru. And while cases in people in the US might be rare, plague outbreaks in animals, especially the rare black-footed ferrets, are devastating. So devastating, in fact, that plague is the top contender for biggest threat in the category of black-footed ferret conservation. The beautiful, sleek black-footed ferret was once thought to be extinct until biologists found a single population in Wyoming in 1981. Since then, thanks to captive breeding and reintroduction, numbers have climbed, but they're still endangered and at major risk of extinction from the plague. But how do you find, prevent, or even treat hundreds of animals hiding in burrows across thousands of acres in the plains of Western North America? The answer might come down to peanut butter and paintball guns. Now, before we dive in, just a quick note about plague. The disease is caused by the bacterium Yersinia pestis, and in wild animals is mostly spread by fleas or by eating infected animals. And when we're talking about plague in wild animals, you might hear biologists calling it sylvatic plague. It's still the same disease. Sylvatic just means of the woods and refers specifically to plague in rural wildlife as opposed to in humans. And anywhere plague occurs in animals, if there is a vector or an organism that can carry and transmit the disease from one animal to another, it can also be passed on to humans. Although there is currently no approved vaccine for people in the US, there are ways to protect black-footed ferrets. Ferrets that are bred in captivity are all injected with a vaccine that uses proteins or antigens from the Y. pestis bacterium. That's great at bolstering their immune system against the disease, but there's a problem. These ferrets aren't the only animal at major risk from plague. Black-footed ferrets rely almost entirely on prairie dogs for their food and use prairie dog burrows for their shelter. So if those animals are wiped out, so are the ferrets. And when prairie dogs become infected, up to 99% of the population can be lost in a single bout. So the conservation effort for black-footed ferrets is as much about vaccinating and protecting prairie dogs that live in the ferret's habitat as it is about the ferrets themselves. And it's not just about keeping the prairie dogs alive, but also keeping them disease-free so they don't pass on the bacteria when they're eaten. In 2004, a team of scientists led by epidemiologist Tony Rock developed one of the first edible vaccines against sylvatic plague from a modified raccoon pox virus. But it only protected prairie dogs who had been infected with plague in about 55% of cases. Later in 2010, Rock and colleagues tweaked the vaccine slightly to include another gene from the virus. This time, 94% of the vaccinated animals survived. Now you might be wondering, how do you get prairie dogs interested in chowing down on plague vaccines? Well, Rock and her team wondered that too. So they tried a bunch of different flavors that might entice the little creatures. Some of the first baits, including the ones used in the 2004 and 2010 studies, were made of sweet potato because prairie dogs regularly ate vegetables as part of their diet in captivity. But when it came to deciding the final bait flavor, the procedure was pretty straightforward. The team put out baits in the lab that had been flavored with either peanut butter, sweet potato, blueberry, or not flavored at all. Researchers then let the prairie dogs loose on them and watched to see which baits were gobbled up the fastest. And peanut butter was the go-to, which Rock said is unsurprising since lots of animals, including rodents, love peanut butter. Now, while they might taste appetizing, these baits don't exactly look appealing. They have to be dyed blue so that they can be easily spotted out in the wild. And once everything is mixed together, the baits are frozen to keep the vaccine itself in working order. So biologists had their vaccine and they had a way to feed it to the animals, but now they needed a way to actually get the bait to the animals. Now, the simplest way, of course, would be to just walk around spreading bait out like lawn seed. But they needed to drop 50 baits per acre and spread them out evenly in a 9x9 meter grid over thousands of acres. Hand drops just weren't gonna cut it. So in 2016, the World Wildlife Fund, the US Fish and Wildlife Service, engineering company Support XXL, and drone manufacturer Model Avionics teamed up to come up with a more high-tech approach. They developed and then tested 3 different robotic vaccines vaccine bait delivery systems. One was a drone that would drop baits one by one, the second was an ATV that again dropped single baits, and the third was an ATV that could drop 3 pellets at a time. That same year, the team tested their designs on 1,200 acres of land in UL Bend National Wildlife Refuge in Montana. Although the ATVs and drones were much more efficient and less pricey than hand-doling the baits out, the lumpy baits sometimes jammed up the chute as they thawed. After a few engineering tweaks, the team came up with their ultimate design, a triple shooter that could be mounted on either an ATV or a drone. Imagine basically like a paintball gun crossed with a gumball machine. Using a trigger that is synced to a GPS, the device pops 1 pellet out underneath it, while at the same time shooting a bait 9 meters out on either side. And you can even double up on the dispensers and have one person drive an ATV and another fly the drone for even more efficient pellet shooting power. In 2017, the U.S. Fish and Wildlife Service used the 2 dispensers to distribute vaccine baits across 7,000 acres in prairie dog habitats in Montana, Colorado, and South Dakota. Now, the team says there are still improvements they want to make. The device does still sometimes get jammed, for example, but overall, it's a pretty good system. Unfortunately, though, oral vaccine baits are not as effective as scientists would like. The vaccine works well in the lab, but in the wild, it's a different story. A field trial by Mark Matchett and colleagues in 2021 concluded that the number of vaccinated prairie dogs surviving until the next year wouldn't be enough to sustain a black-footed ferret population. They think the low success rate comes down to timing. See, autumn is the best time to distribute the bait, since food quality slowly drops and prairie dogs would be motivated to snack on some bait on the side. But even though 99% of the prairie dogs the scientists sampled had eaten at least one bait, come spring, a whole lot of prairie dog pups are born, meaning around half of the population ends up not being vaccinated. Which means, as cool as the paintball shooting peanut butter vaccine balls are, they are just one part of a conservation plan to control plague. Capture and release, as well as using insecticides to kill the fleas that spread plague, are also very important. Plague will likely always be there, but by combining research and technology, maybe it doesn't have to be so much of a plague.
A horde of leeches approaching you in the forest might not be the most welcome sight. That is, unless you're a conservation biologist. See, these little vampires carry something invaluable to these scientists: blood. And not just any blood. The blood of many different animals. It's important for conservation biologists to know which creatures live in an ecosystem, so they conduct biodiversity surveys to get as accurate a wildlife census as possible. But unlike a human census, scientists don't have as simple a method as posting at forums. Some survey methods are expensive, some are invasive, and others are downright impractical. But with a belly full of blood, leeches might just be able to survey the ecosystem for us. Now, these aren't the typical leeches that live in ponds that you might be picturing. There are actually terrestrial leeches too. They live in humid tropical climates and also enjoy a tasty blood meal, which can remain in the leech's system for months. And in 2012, a study was published showing that leech blood meals can be used to identify wildlife. This works because leeches store their blood meals in their systems for a long time. And scientists can extract the DNA from the blood cells and identify it through a polymerase chain reaction test, or PCR for short. PCR works by recreating the process of DNA replication in a test tube. You might have heard of it before in the context of the COVID-19 pandemic. It's the same test. Scientists mix a DNA sample with some enzymes that act like photocopiers of DNA, and molecules that tell those photocopiers exactly what genes to copy. Several rounds of this replication result in many copies of that specific gene, enough to be easily detected by a machine or other lab tests. This can help scientists look for the DNA of specific animals. For instance, if a scientist wanted to check if a tube of blood came from a goat, they can mix it with molecules that will only copy goat-specific genes and run a PCR. If there's any goat DNA in the blood, PCR will make it obvious. Long story short, this means that the leeches can act as squirming tubes of DNA samples with generous expiration dates. And this got conservation biologists excited. When researchers want to study an ecosystem's biodiversity, they need to survey the creatures somehow. That can be done by capturing the animals, but being caught and poked at by scientists can be stressful. Researchers can also observe animals that wander by trail cams or collect samples that they leave behind, like feathers, hair, or dung. These methods are much less invasive, but some animals are fickle and elusive, so scientists may never see hide nor hair of them. So, these researchers thought, if leeches have a varied diet, they could be a welcome alternative to catching animals or hunting down their poop. And one study published in 2018 by Michael Tesler and colleagues looked into the bellies of leeches in Bangladesh, China, and Cambodia to see what was there. Turns out, it was a lot of different kinds of blood. Genetic analyses of the blood meals revealed DNA from 29 different species, including bats and birds. That's a pretty convincing argument for inviting leeches into your lab. One of the scientists involved in the 2018 study, Sarah Weiskopf, found its results very promising and was curious how leech meals held up to other, more traditional survey methods, specifically trail cams. Trail cams, or camera traps, are the go-to for tracking wildlife in tropical rainforests, but they're expensive and take forever to do their job. So, she led another study, also published in 2018, that pitted leeches against cameras in the forests of Bangladesh, and the results were complicated. The cameras found 14 more species than the leeches had, but the leeches found species that the trail cam had missed. Also, the leeches were collected over 4 days compared to the 9 months that the cameras ran, and the leeches were over $20,000 cheaper. So, collecting leeches isn't the perfect wildlife survey method to end all methods, but it's pretty darn good. And in the world of science, there's often no single method alone that can perfectly answer the questions that researchers aim to address. Our best bet is an arsenal of procedures that are pretty darn good. And while they might not be able to hold a test tube or fit into a lab coat, leeches might just earn a spot on the conservation biologist's lab bench. Everything changed when the goats arrived. Before, giant Galapagos tortoises acted as their island's gardeners, thinning overgrowth, dispersing seeds, and basically maintaining balance in the ecosystem. And that was good for everybody living there. But the delicate ecological situation of the Galapagos Islands was upset when pirates and whalers paid them a visit in the 1800s. While their stay was temporary, their stowaways, feral goats, made themselves at home permanently and brought environmental destruction with them. Conservation scientists tried to remove the threat, but their first attempt wasn't effective enough. What they needed was a bit of creativity, and what they came up with was a way to turn goat against goat. Now, these goats might not have been a problem if they were a good fit for the ecosystem, but they weren't. They were invasive to the Galapagos, meaning they weren't native to the islands. With no natural predators to keep them in check, their presence spelled trouble for the habitat. These goats were fast, hungry, and destructive to the island vegetation, much like disrespectful teenagers tearing through your kitchen cupboards and leaving a mess for you to deal with later. But unlike us, who can clean up and restock our snacks at the grocery store the next day, the Galapagos can't bounce back so fast. The goats' ravenous rampage disrupted the delicate balance of the island's ecosystem. The plants couldn't grow fast enough. And as you might guess, the tortoises weren't fast enough either. Though the gentle giants act as good stewards for the island's vegetation, they lead slow, peaceful lives. Clocking speeds of 0.2 miles per hour and sleeping 2/3 of each day, there was no way that they could compete with a horde of fast and hungry goats. Their population dwindled. It's important to keep in mind that these particular giant tortoises are endemic to the Galapagos, meaning they're found nowhere else on Earth, along with many of the other species living on the islands. If their habitat disappears, they do too. And the tortoises weren't the only part of the islands affected by the goats. Overgrazing altered the balance of island vegetation, leading to significant soil erosion. Eventually, the locals said that the goats had caused enough trouble. But there was no obvious or easy solution. The problem was larger than simply shepherding a small herd onto a boat off the island. Conservationists debated introducing predators to take out the goats, but that had the potential to bring even more issues. So in 1997, Galapagos scientists and land managers launched Project Isabella, named after the island on which it took place. The project was simple in concept. Sharpshooters flew around Isabella Island on helicopters and eliminated any goats they found. While the effort was ruthless, it was effective, at least at first. Except there was one little problem. Some goats were really good at hiding. So good, in fact, that they outsmarted the hunters until the conservationists were back to square one. It became clear that all the goats needed to go. With their ability to bounce back, it was either the sensitive Galapagos ecosystem that had to disappear, or the goats. It seems that the situation called for a more cunning solution. Introducing Judas goats. Yes, named after that Judas. About 900 goats were sterilized and given tracking devices. Then they were turned loose onto the islands. Goats are herd animals, so they band together. The Judas goats were no exception, finding new companions upon release. And just like how traitorous Judas betrayed his friend Jesus, the tracked goats led their brethren to their own demise. Scientists have been using radio trackers to track animals since the 1960s. These trackers are worn by the animals, revealing their location by transmitting signals to the researchers. Radio tracking has been very useful in conservation because we can glean a lot of good information from an animal's location, its migratory patterns, personal habitat use, and social behaviors, to name a few uses. In this case, radio tracking came in handy for hunting down these Judas goats and their friends. The shooters would follow the signal and eliminate all but the Judas goats, leaving them alive to find other goats to track down. Then the cycle would repeat. And this worked. Not even the smartest or stealthiest of goats saw it coming. The plan was so effective that northern Isabella was declared goat-free by 2006. That's a huge feat considering the goat population had reached roughly 100,000 in 1997. After this, balance began to return to the island. The vegetation grew back, and the Galapagos tortoise population grew by around 16,000 individuals across all the islands, which is a lot of big tortoises for such tiny islands. The issue of invasive and introduced species is much larger than the goats or the Galapagos. They are the second largest threat to biodiversity today, only beaten out by habitat loss. And their impact is much worse on islands, as miles and miles of water are pretty effective at isolating an ecosystem. But sometimes this problem can be solved. Some solutions are more effective than others, but with creativity and adaptability, conservationists can work to nudge our world's ecosystems back into balance. Since the late 1980s, marine biologists have been shooting whales with crossbows to get samples of their skin and blubber, which sounds pretty medieval, but there wasn't a better way to get at the genetic material and fat the researchers needed to study things like their genealogy and how polluted their environment was. It can tell them a lot more too, and all this information can help in conservation efforts. DNA and a genealogical history, for example, allows researchers to see how populations are changing over time due to things like whaling or climate change, as well as more current events like which males are fathering calves. And traces of pollutants, like pesticides, stored in the animal's blubber, tells them about potential threats to whale survival. But this stab and see method wasn't exactly what you'd call friendly or low stress for the whales or the researchers. Plus, biologists often want to study what happens to individuals or populations over time, which means shooting them more than once. And no one wants to turn these beautiful creatures into Swiss cheese. So in the last 15 years, researchers have turned to a different kind of sample, snot. And the way they're getting at it is not only much more whale-friendly, but also pretty high-tech. Now, collecting a sample is pretty standard practice in biology. A researcher will take a little piece of something, whether that's a chunk of coral or some tissue or blood from an animal. But getting that sample is rarely easy. If you've ever tried to chase after your dog with something it's not supposed to have in its mouth, you'll know what I mean. It gets even trickier when that animal lives in an environment we're not really made to go into, and the animal itself is 18 meters long and weighs more than 60 metric tons, like whales, which is where the giant biopsy dart on a crossbow method came from. But in 2011, a new idea hit researchers right in the face. Literally. While hanging out on a boat following whales, a researcher named Ian Kerr was showered in buckets of water and smelly mucus that had been shot out as part of the whale's spout. And he thought, why not collect this stuff instead? It might sound kind of gross, but whale snot, also called whale spout or sometimes whale blow, is full of clues that can tell researchers about the whale's health or life stage. These are things like cells from the surface of the lungs or mucus, proteins, fats, bits of DNA or nucleic acids. Kerr was also a fan of drones, or uncrewed aerial vehicles, and thought the tech could be used to collect this whale snot as well. The idea of a flying snot robot was born. Each drone is kitted out with 6 petri dishes that collect the goo. Researchers will sit on a boat hundreds of meters away from the whale, launch the drone 20 meters into the air, and hover over a whale and collect its exhaled breath when the time is right. And it's great because the aerial vehicles allow scientists to stay far away from the animal and not stress them out with their presence. Plus, the machines are fast and maneuverable, perfect for catching up with a speedy animal or one that only pops up occasionally, like a surfacing whale. And several research groups have already put their own snot-collecting robots to good use. For example, in a 2018 study, Gemma Gijan and colleagues sequenced the virus genomes found in eastern Australian humpback whale spout to learn more about how viruses take hold when the animals are stressed and how they're passed around. And in 2021, a marine mammal professor named Shannon Atkinson and her team used whale spout to assess the health of humpback and blue whales in Alaska, including measuring some of their hormones and screening for diseases. This could help with conservation and management of these populations in the future. And as a part of a 2024 study of whales in Canada, Aideen O'Mahony and colleagues were able to reconstruct the genomes of humpback and fin whales using DNA found in whale snot, gaining insights about the genetic diversity of the population. Plus, other scientists have started using these same drones, and drones in general, to further expand what they know about these majestic creatures. For example, some drones can put trackers on whales that measure their movement and location. This helps the scientists learn even more about the whale's feeding behavior, movement and migration, or how their swimming changes due to things we humans do, like drive boats close by. One paper from 2023 even described how drones can just fly on by and plop a tracker with little suction cups attached to the whale's back, pretty much without it even knowing. And by combining drones with another hot topic bit of tech, deep learning algorithms, researchers can take super high resolution images of whales from above and calculate precisely how big they are to know how healthy individuals and pods are. But of course, conservation tech doesn't stop at drones. There's also special microphones or recorders that can be placed in whale habitats to record their calls and get a sense of who's around and what they're doing, be it looking for food or socializing. And sometimes planes are used to track the path whales take while migrating, and maybe even see if there's any dangers along the route. All these studies build a picture of how well each whale is doing, what's stressing them out, whether it's ship noise or pollution, and how the population might change. And that information is vital in helping conservationists come up with strategies to protect these majestic animals without having to get out the crossbows.